Today, we are diving deep into the cell's primary power generator with a step-by-step guide to the Krebs cycle. Think of this process as a finely tuned metabolic engine. Its main objective is to systematically strip carbon molecules apart to extract the vital energy required to keep you alive. Let's break down exactly how this machinery operates. Before we start the engine, let's look at where this happens. If we zoom into a standard animal cell, we find the mitochondria, and deep inside their inner fluid, the mitochondrial matrix, is where the Krebs cycle runs. There is one unique exception, an enzyme called succinate dehydrogenase, which is actually physically bolted directly to the inner mitochondrial membrane. The ultimate goal here is to take acetyl-CoA, which comes from the breakdown of carbohydrates, lipids, and proteins, and completely oxidize it into carbon dioxide. This process releases high-energy electrons that will later power the electron transport chain. Remember, unlike linear pathways like glycolysis, this is a perfect, continuous circle. To ignite the engine, we have to prepare our fuel through a gatekeeper reaction. We take pyruvate, a 3-carbon molecule from glycolysis, and introduce it to a massive enzyme machine called the pyruvate dehydrogenase complex, along with coenzyme A and NAD+. This enzyme triggers an irreversible reaction called oxidative decarboxylation. It chops off one carbon atom, releasing it as carbon dioxide waste, and generates one molecule of NADH. What remains is a highly energetic 2-carbon fuel cartridge known as acetyl-CoA. Now the engine officially starts running. In step 1, the citrate synthase enzyme takes our incoming 2-carbon acetyl-CoA cartridge and fuses it to a permanent 4-carbon carrier molecule called oxaloacetate. This condensation reaction uses a water molecule to build a brand new 6-carbon compound called citrate. With the connection made, coenzyme A is released back into the matrix, free to fetch another round of fuel. With our 6-carbon citrate ready, the engine performs a quick structural rearrangement in step 2. An enzyme named aconitase temporarily removes a water molecule and then immediately adds it back, but in a slightly different position. This transforms citrate into its direct molecular isomer, isocitrate. The total carbon count remains firmly at 6, but the molecule is now perfectly reshaped for the chemical extractions ahead. Step 3 brings our very first major energy payoff. The enzyme isocitrate dehydrogenase steps in to snip a carbon atom straight off the backbone, which is immediately exhaled as carbon dioxide waste. Breaking this chemical bond releases high-energy electrons, which are captured by NAD+ to create our first payload of NADH. Because we lost a carbon, our molecule shrinks down from a 6-carbon structure to a 5-carbon compound called alpha-ketoglutarate. As a quick memory aid, remember the acronym KEAM, ketoglutarate, isocitrate, and malate are the specific steps that yield NADH. In step 4, the engine duplicates its previous strategy using the alpha-ketoglutarate dehydrogenase complex. Another carbon atom is sacrificed and released as carbon dioxide waste, while a second payload of high-energy electrons is packed safely into another molecule of NADH. To stabilize the highly reactive 4-carbon fragment left behind, the enzyme temporarily tethers it to a fresh coenzyme A molecule, creating succinyl-CoA. An interesting side note, the 2 carbons lost as waste up to this point actually originated from the original oxaloacetate carrier, not the new fuel cartridge we just brought in. Step 5 serves as a highly unique factory station managed by succinyl-CoA synthetase. Here, the temporary coenzyme A tether is cleaved apart. The sheer energy released from breaking this high-energy sulfur bond is immediately harnessed to slam an inorganic phosphate group onto GDP, generating a molecule of GTP. Since GTP is chemically equivalent to ATP, this represents our first direct harvest of cellular energy currency. We are now left with a clean, unattached 4-carbon molecule called succinate. Moving into step 6, we reach the membrane-bound station. The enzyme succinate dehydrogenase, which, as a reminder, is physically bolted to the inner mitochondrial membrane, oxidizes succinate into fumarate. Because this particular step releases slightly less energy than the earlier oxidation steps, it isn't powerful enough to reduce NAD+. Instead, the enzyme transfers these electrons to FAD, reducing it to the medium energy carrier FADH2, which plugs directly into the neighboring electron transport chain. Step 7 functions as a quick preparatory adjustment station. An enzyme called fumarase adds a water molecule directly across the carbon-to-carbon double bond of fumarate. This straightforward chemical hydration changes the structure completely, converting it into L-malate and setting the stage for our final energy extraction. We have reached the 8th and final step, resetting our metabolic engine. The enzyme malate dehydrogenase performs a final oxidation, transforming L-malate back into oxaloacetate. This last chemical breakdown allows us to extract one final payload of energy, reducing NAD+ into our third and final NADH molecule. Our permanent 4-carbon carrier is now completely restored to its original state, standing by and waiting for the next molecule of acetyl-CoA to arrive so the cycle can turn again. Let's review the entire energy ledger to see exactly what this engine yields. For every single turn of the cycle processing one acetyl-CoA, we harvest 3 NADH molecules, 1 FADH2, 1 direct GTP or ATP, and release 2 molecules of carbon dioxide waste. However, because a single molecule of glucose splits into 2 pyruvates during glycolysis, the cycle must turn twice per glucose. That brings our total yield per glucose molecule to 6 NADH, 2 FADH2, 2 direct ATP equivalents, and 4 carbon dioxide molecules. While making only 2 direct ATPs might seem low, the massive potential stored inside those NADH and FADH2 carriers will go on to yield over a dozen more ATP molecules during oxidative phosphorylation. Finally, let's step into the control room to see how the cell regulates this engine. To slow things down, the cell uses a process called product inhibition, which acts as the brakes. When concentrations of ATP and NADH are high, it signals that the cell's battery is completely full, strongly shutting down pyruvate dehydrogenase, citrate synthase, and key dehydrogenases. Additionally, succinyl-CoA acts as a negative feedback loop to block its own overproduction. Conversely, when energy is low, the accelerators kick in. High levels of ADP directly activate isocitrate dehydrogenase to speed up the cycle. Furthermore, during heavy physical activity like muscle contraction, calcium ions flood into the mitochondrial matrix.